So it's a great pleasure to welcome all of you here. We have a nice speaker today. It is Mazin Jouda. He received his bachelor degree in electrical engineering and Master of Science in microsystems engineering from the Institute of Microsystem Technologies at the University of Freiburg in 2011. He completed his PhD with distinction in 2016 at the Institute of Microstructure Technology, Karlsruhe Institute of Technology, where his research focused on developing innovative electronics interfaces from massively parallel MRI-phased array systems. Since 2017, Dr. Jouda has been a group leader at the Institute of Microstructure Technology in Karlsruhe. His group focuses on advancing hardware and signal processing solutions for MRI and spectroscopy. So, dear audience, please hold your questions for Dr. Jouda until the end of the talk or submit them in the chat. And with this, Dr. Mazin Jouda, stage is yours.
Yeah, thank you, Andrew, very much for this very kind introduction. And good evening, everybody, and welcome. It's my great pleasure to be invited to this seminar and to give you an overview and some highlights about our research activities. As Andrew already mentioned, I'm a group leader at the Institute of Microstructure Technology at Karlsruhe Institute of Technology. And in my group, we focus mainly on developing microsystems and hardware, as well as methodology solutions to advance NMR spectroscopy and magnetic resonance imaging. This is the outline of my talk today, and significant part of our research activities is driven by the motivation to having high-throughput NMR spectroscopy. Therefore, we focus a bit more on spectroscopy. However, all the technical and method advances that I'm going to present today can be applied equally and straightforwardly to magnetic resonance imaging, as I will show a couple of examples through the talk. Towards high-throughput NMR spectroscopy, we have to address several technical and method challenges. Therefore, I'm going to talk today about parallelization, for example. I'm going to talk about miniaturization and optimization as important tools to achieve parallel spectroscopy. Also, I will highlight the use of AI-based shimming, so using deep learning, namely, to speed up the shimming process in spectroscopy, and using also new ways of detecting the signal, namely also using digital lock-in detection as an alternative to conventional analog lock-in detection, which opens up also new opportunities to acquiring multiple nuclei. And I will talk also about microcoils as an important way of miniaturizing the coils and allowing measurement of multiple samples simultaneously. And finally, I will also highlight some advances on using microelectronics to tremendously reduce the complexity and size of the NMR and MRI electronics. So spectroscopy is a very powerful analytical tool that gives spectacular insights about the molecular structure as well as the dynamics of materials with atomic resolution in a completely non-invasive and non-destructive way, which makes it very widely applicable in many fields. However, this technology suffers from a major drawback besides, of course, the low sensitivity, which is the low throughput. So even if you have the most advanced NMR system, you will not be able to measure more than one sample at a time. And to understand where this limitation comes from, we have to look inside the NMR system or the magnet where we have the sample. And for spectroscopy, we need the field homogeneity inside the sample to be extremely high, talking about variations only in the order of few parts per billion, which can only be achieved by a set of coils called the shimming coils, and that represent the different terms of spherical harmonic expansion that is typically used to decompose the inhomogeneous field inside any sample. So as you see here, those, by definition, those spherical harmonic terms or shim profiles are all symmetric around one center point on which the sample should be centered. Therefore, if we want to increase the throughput of our NMR experiment and we want to add, for example, multiple detection sites or multiple detectors with addressing multiple samples, then by definition, a single shimming system or shimming set will not be able to shim multiple samples that are located at different spatial positions simultaneously. So that's one of the major barriers. I start with the second one, which is shimming. And the other one is the RF coupling between the channels. So these are detectors that are typically operated at tens to hundreds of megahertz, depending on the magnetic field. And being close to each other increases the chances that there is RF coupling and crosstalk between the detectors so that one detector can see the neighboring samples as well. Add those challenges, we introduced localized shims as a way that each sample can be equipped with its own shim set so that it can be shimmed independently of the other samples. And the first prototype to validate this concept is this array here that has two NMR cells, each of which has an RF detector that addresses the 5 millimeter sample. The RF detector is wrapped around the 3D printed scaffold, and there's another support structure of the 3D printed scaffold on which we wrap the localized shim set. This array, we use this preclinical imaging magnet to validate this concept because of the relatively large available space inside the magnet. And we limited, for the first prototype, we limited the number of shimming coils per cell to 3 shims addressing the linear first order shims, XYZ. So the RF coil was built. We used the saddle coil architecture with 2 loops to build the RF detector, and has to be tuned at 45 megahertz, corresponding to the 1H Larmor frequency at this magnet. And the orientation of the RF coils and the cells was optimized so that we have an orthogonal orientation so that the RF coupling between the 2 cells is minimized, as can be seen here by the S parameter measurement of the 2 cells. We also tried to make the 2 coils far from each other, so the 2 cells far from each other, still close enough so that they fit in the kind of relatively good field of the magnet. Similarly, the shim coils for each cell were optimized using finite element method-based simulation. So we used COMSOL multiphysics to do the optimization. And the optimization basically targeted the geometry of the shim coils so that their profiles are as linear as possible along the X, Y, and Z. It was a parameter suite optimization to achieve the desired profiles. And again, they were also fabricated and manufactured using multi-layer flexible PCB technology. And after that, they were wrapped around the 3D printed support structures and mounted on the PCB and soldered to the substrate. After managing to shim both samples, we could get to the best achievable line width for both channels at the same time. These figures show the NMR spectra of two water samples with and without when the localized shims are turned on and turned off. And as you can see here, turn off the localized shims, you can see that the field inhomogeneity, the respective sample is too high that you can see the signal. And this also emphasizes the point that within one shim system, you can't shim both samples at the same time. So you need those localized shims to be able to achieve good homogeneity in both samples simultaneously. Okay. The shimming process was a bit tricky here because in this case, we have two cells, each is equipped with 3 localized shims. So in total, we have 6 localized shims. And we have additionally 3 shims from the commercial MRI system. We call them global shims. So in total, we have 9 shim currents that we have to optimize. And the strategy that we use to do that is first we turned off the localized shims and then try to use the global shims to shim only sample 1 to the best possible line width. And as you can see, when sample 1 is okay, then the shim the field inhomogeneity in sample 2 is high and sample 2 is not good anymore. And then we reverse this process, so we can try to use the global shims to correct the field in sample number 2. And again, you see that sample 1 now is not good anymore, emphasizing the point that we need localized shims to be able to do both at the same time. And after that, we just recorded the shim values for the 2 scenarios, and then we interpolated them. So we put the shim into an intermediate state where both samples are okayish, but they are not yet perfect. And after that, localized shims could take over. And by correcting only the localized shims afterwards, we could get the 2 spectra to the best possible achievable line width at the same time. And here are some examples of spectroscopy experiments of 2 samples simultaneously. So every column shows an NMR experiment of 2 different samples. And as I mentioned before, this is a preclinical imaging magnet, so it's not optimized to do spectroscopy. However, we could resolve the chemical shifts there, but the J-couplings were not yet resolvable. And the interesting thing here is that even with doing averaging there, we could not see any observable crosstalk between the NMR spectra. Okay, then we also use that setup to do hyperpolarization experiments of like simultaneous hyperpolarization of 2 different samples. We used Zebra hyperpolarization. So here's the kind of experimental setup where we have perhydrogen and we have 2 samples, the samples that we want to hyperpolarize. We have pyrazine and pyridine. And then we pump them through this tube-in-tube reactor, which is placed in the polarizing electromagnet. And after hyperpolarization, we pump the hyperpolarized samples to the NMR detection cells. And of course, because here the flow plays a role in how much enhancement we can achieve, we needed to optimize the flow to achieve the maximum enhancement, and we ran a series of experiments where we increased the flow rate and recorded the NMR spectra, and then from that we could figure out that the maximum enhancement is achieved when we have a flow of 6 milliliters per minute. And to quantify the enhancement in the signal, we ran imaging experiment on a reference sample that is not hyperpolarized. And then we hyperpolarized pyrazine sample. And by calculating the protein concentration for both and the number of averaging, we could measure enhancement of around 2000 in signal intensity. We could also run parallel hyperpolarization experiments, and with all the different scenarios of turning on and off the hyperpolarization for both channels. So here, both are at thermal polarization, and for this low concentration, we could not see anything with a single shot experiment. And after that, we could turn the hyperpolarization. And again, the interesting thing here is that even with hyperpolarization, and we could not see any observable coupling between the two channels. And now we have to talk about another topic, is if we want to go further with high throughput, if we want to pack more and more detectors, increase the number of detection cells within the array. Of course, size, limited space inside the magnet will be a major barrier, so we need miniaturization. We also need to think of new detector topologies that are more shielded than saddle coils, for example, so they can allow more densely packed arrays without raising the risk of having crosstalk between the detectors. And also we have to optimize the entire cell as a unit so that we could achieve better and higher spectroscopic resolution without necessarily increasing the number of shimming coils. This is a recent publication where we introduced kind of a simple and cheap and very reliable way of making microdetectors. The idea here is to use extrusion of silver ink on a glass capillary to build micro coils. Here's the kind of the printing setup. We have the printing head where we put silver-based ink, and then we apply pressure here so that we can control the volume of the ink that comes out of the nozzle. And then we have an XYZ stage together with a rotation capability so that we can turn the capillary while we are extruding the ink on it. And here are some examples of printing silver based microcoil in a glass capillary of 1.5 millimeter diameter with different number of windings and different pitch sizes. After printing, we have to center the silver. So we put the printed microcoils in an oven at 220 degrees for 60 minutes for the sintering. And from this laser scanning microscope images, you can see that we can achieve wire thicknesses down to 60 micrometer with a height of 9 micrometers. Of course, that can be probably pushed even further by optimizing the pressure and the printing parameters and the speed by which we can move the XY stage and the rotation here. This microcoil was tested in both imaging and spectroscopy experiments. So he's in a spectroscopy experiment, a one-shot spectrum of ethanol sample that was put in this capillary. And here's an example of an imaging experiment when we fill this capillary with DI water, and we get a sagittal slice and an axial slice, and then we put a thin Teflon tube inside the capillary. Interestingly, we could achieve within like a small number of averages, like 32 averages, we could achieve in-plane resolution of down to 78 micrometers. Another technical advancement that is necessary if you want to do high throughput spectroscopy is this coil that we published a couple of years ago. Here we introduced a new coil. We call it butterfly stripline. So a conventional stripline that's widely used in NMR spectroscopy looks like that. So it has two planes, the ground plane, the bottom plane that's called the ground plane, and the upper plane, which is the signal. And it's a plane that has a narrow part, now normally called the sensitive part, where the sample is to be detected because of the higher current density. This coil has a lot of advantages, but has a major disadvantage that it's a single loop coil. So in principle, the only way to increasing the sensitivity of this is to make the strip line wider. And sometimes you have space constraints in the magnet, so you can't make this part wider, which is a limitation then. And to solve that, we introduced a new way of coils. We call it the butterfly strip line, where we replace the sensitive part of the strip line, the single loop coil, with a figure 8 or butterfly coil, where the number of windings can be increased beyond one loop. Increasing the number of windings definitely increases the sensitivity, as it can be seen here. So we have this figure here shows the notation spectrum comparison of the conventional strip line here and the butterfly strip line, and we can see that the new design offers a factor around a factor of 2 enhancement in the signal. The other interesting feature of this new detector is its inherent shielding that's better than the conventional strip line. As you can see here from the console simulation, we see that the butterfly strip line, the new one, is more sensitive to the sample, but at the same time, its stray B1 field is smaller than the conventional one, making it a better candidate for making NMR arrays. Okay. Another interesting feature of the new detector is its ability to suppress the unwanted signals outside the sensitive part. As you can see from this simulation here, this profile shows that the new butterfly strip line is better in terms of suppressing the unwanted samples outside the sensitive region. And here are some measurements of the sample that is purposefully placed outside the sensitive region, and we can see that the conventional strip line can detect that with more sensitivity than the new strip line. And those as parameters show the coupling when we have an array of two detectors when they are placed side by side or back to back, and for different distances between the detectors. And in all cases, we see that the new design offers at least 10 dB more shielding or more decoupling when arrayed. Okay, now we come to the optimization. And previously, when I showed the NMR, the results of the parallel NMR spectroscopy, we could not achieve J-coupling resolution. The reason is that we kind of optimized the shims and the RF coil individually. So the RF coil was optimized for sensitivity, whereas the shim coils were optimized for the desired field profile. However, a key thing to achieving a smart shim is to optimize the entire NMR cell as a unit. And this is what we did here. So in this case, we decided to use a 1 millimeter capillary as a sample holder, so that we have enough sample, so it's not too large. to account for a lot of inhomogeneities in the field, but not too small to also have too little signal for the shimming. And then we try to optimize the thickness of the RF detector, the wire thickness of the RF detector, so that we minimize the inhomogeneity inside the magnetic field inhomogeneity inside the sample. This is a console simulation showing the effect of the wire thickness on the B0 field inside the sample. And as you can obviously see here that for a thinner wire, we have less inhomogeneity. And if we plot a histogram of the field distribution reflecting the kind of the anticipated line width that we would achieve for this scenario, we will see that we have the least inhomogeneity for the thinnest wire. But the other interesting thing is that if you look at this blue curve, you see that it's more or less kind of symmetric, which suggests that this inhomogeneity could be corrected by only linear shims. And once we are happy with the optimization of the RF coil, we designed a shim set of linear shims XYZ that is to be placed around the RF coil like that. And then we use console optimization to optimize the geometry of the shim coils to achieve linear profiles inside the sample. And we ensured in the optimization that the profile is along one axis is the same regardless of the offset along the other axis. To ensure that we have an optimized shimming profile. And you see here the experiment. We did an experiment using this NMR cell, and you see without the shims here, we didn't use any global shims. So the shims of the commercial magnet were turned off, and only the localized shims were active. When we turned all the shims off, that's the line width that we could achieve. And with only 3 linear shims, we could resolve the J coupling, so we could get down to 4 Hertz line width. Now, the other topic that I want to talk about is a methodology. And now you see if we manage to solve the shimming problem, we could now run an array of NMR detectors with efficient shimming, and we could achieve high spectroscopic resolution in an array. But the other problem that arises now is the shimming process itself, because the more NMR cells we will have in our array, the number of shims will increase exponentially, and the shimming process will become extremely time-consuming. The shimming itself the process itself is time consuming because we have to find the correct currents for the shim coils, or the correct weights of the spherical harmonic turns, and theoretically those coils are kind of orthogonal to each other. However, practically they have some slight non-orthogonalities between them, making the shimming process quite time-consuming because of its iterative nature. Commercially, there are optimization algorithms that are applied in commercial systems to do the shimming, like the simplex or the gradient-based shimming. However, they are still time-consuming solutions, and they cannot be a good candidate if we want to do massively parallel NMR spectroscopy. So we need new ways of doing the shimming that are faster. And one way was to use deep learning to speed up the shimming process. So in this first prototype, we thought the following: if we have a distorted spectrum, so unshimmed spectrum like that, and we could have 3 copies of this unshimmed spectrum. can we use this set of spectra, apply it to a deep learning model that can that can predict correct shim currents so that we can get a better line width. That was kind of the initial hypothesis from which we started. We said we will limit our model, our first model, to the linear shims only. So we'll just use XYZ shims, and we will turn all the other shims off. But first, to be able to train a model, we need a data set. We need a large data set. So we decided to use this NMR, this benchtop NMR spectrometer from Magritech to collect a large data set of spectra, around 9,000 NMR spectra. And we decided to do the sampling in a uniform way. So we start from the shim, the very correct NMR spectrum, then we apply systematic distortions on the shim current of XYZ. And for every sample, we do 3 copies around that sample with a fixed offset along the X and Y and Z. And then we have an entry. And then we repeated that 9,000 times so that we have a big data set to train our deep learning models. And we used 2 different architectures. So we used convolutional neural networks and one with only 1 weak learner architecture, and the other with an ensemble of weak learners, so that we at the fully connected layers, we just look and select the best prediction from all these learners. We trained the neural networks, and then we tested them on a validation and data set of 100 spectra. And we looked on both how much enhancement we get in the line width and how much enhancement we get in the amplitude of the spectrum. And we compare that to the simplex algorithm that is already applied in the spectrometer. So for the simplex, for 3 shims, it needs at least 10 iterations. And from 10 iterations, it enhances the line width and the amplitude by a factor of 5. For our deep regression, with 1 iteration, 1 acquisition, we get already quite similar to the enhancement of the simplex after 10 iterations. So we get around 4.6 enhancement. And if we add the simplex after that, we can get more enhancement. So we get 15 times enhancement in the line width and the magnitude, telling that, okay, this deep regression model helps the simplex to avoid getting stuck in local minima. And the other model, the deep regression ensemble, is more or less the same performance. So we do not see a significant enhancement between this and that, but both of them, they show that they can in 1 acquisition already get to the same quality as the simplex for 10 acquisitions, and they can both help the simplex afterwards to avoid local minima and get further enhancement. And here are some examples. So of course, the sample we used for training data was water. As you see here, if this is this line here is the spectrum, the unshimmed spectrum, from which the model starts, and it already in 1 shot can predict the correct shim, which is quite close to the optimum that could be achieved by the simplex algorithm. So this is the deep regression. This is the deep regression ensemble, and it's also kind of similar performance. The interesting thing is that also when we tried different samples that were not used during the training, the model was still able to predict the shims quite correctly. Okay, then we tried to advance this further. So we decided to do some changes. So we decided to increase the number of shims and include the z-square shim. And we changed the model so that we used a recurrent neural network where we put some kind of memory in the model, so the model can learn from the previous attempts during the shimming process. And we decided that the model can do a number of random steps. We call them R, and a number of predictive steps. So first, the model can try random steps in the shimming to localize itself in the shimming space, and after R random steps, the model can do predictions. We also changed the way how we collect the training data. So instead of having a uniform sampling for the training data, we decided to use a random sampling. And of course, here we have to optimize because we decided a certain number of random steps and predictive steps. We have to optimize this. We want this to be as small as possible, but still enough to give good predictions. And we don't want to have the predictive and random steps to be too many, so that we lose the speed compared to the conventional optimization algorithms. Therefore, we trained the model with different scenarios for the random steps and the predictive steps. We tried 10 random steps and 10 predictive steps, and we could see that the best performance of the model happens when we have 7 random steps and 2 predictive steps. So the shimming, after we trained the model, we validated it, and the results were quite impressive. The shimming works like that. So this is the shimming scenario. We start from this red spectrum, which is the distorted or unshimmed spectrum. And the model start making 7 random steps. So these are the dotted line. These are the spectra corresponding to those 7 random shim predictions. And after that, the model start doing predictions. So it does 2 prediction steps. And you see immediately it goes from the unshimmed to the almost the optimum shimming condition. Sorry. Okay. And also, we tested this model with a sample that was not used for the training. We used always water for training. Here is spectrum of vitamin B3. And as you see, here's the starting spectrum. That's the unshimmed spectrum with a line width of 4.5 hertz. Then when we apply our enhanced deep regression model that requires 9 acquisitions, 7 random steps, and 2 predictive steps, we already get to 0.51 hertz. If we after that apply a parabola optimization algorithms that's also used by commercial systems and requires 14 extra acquisitions, we can get 0.4 hertz line width. And compared to the simplex that takes 90 acquisitions from here to here, we are not too far. So already with the AI model, we can get 0.5, which is quite close to what the simplex can get in 90 acquisitions. Or if we apply the parabola after the simplex, we in a total of 23 acquisitions, we can still get almost to the same shimming quality. After that, we apply this method to parallel spectroscopy, so at the high field system. So this is a home-built probe that has 2 NMR detectors, and each detector is equipped with a set of shimming coils comprising 6 shim coils, namely the X, Y, Z plus Z square and Z cube and Z to the power 4. Again, we need to collect a large data set for training from the parallel setup, And then after that, we modified the AI model to be trained to be able to predict 12 shim currents simultaneously. And as you can see, here are examples of how this model works. Again, it starts from unshimmed spectrum and then does seven random shimming steps, and after that, it immediately predicts the final shims, which is more or less similar to what we could achieve manually. Unfortunately, there exists no optimization algorithms for such an experiment. It's completely new. We have a parallel setup, and we don't have on the machine an optimization algorithm that can shim this thing. But the AI shows that it has a lot of potential for parallelization. It does not scale as the number of shims, which is the good news, which means that we can increase the number of cells and the number of shim coils, and the complexity of the shimming process will not scale with the same factor. Okay. After that, I'll briefly talk about efficient signal acquisition. Another barrier to scaling up this concept further is the electronics. So if we want to increase the number of detectors, we need for every detector a dedicated hardware to be able to receive and process the signals. And conventional systems or commercial systems like this preclinical imaging system, the signal processing looks like that. So most of the processing is done in the analog domain, like filtering and amplification and demodulation, and only the signal is digitized when it's in the baseband. Of course, this makes these receive chains not very flexible. And if we want to acquire, for example, from multiple detectors, then we need separate receivers for every detector. What we did here is we tried to approach this differently. So we have this digital lock-in amplifier. These are kind of getting very popular nowadays because of the advances in semiconductors and the possibility to have very high speed analog to digital converters. This works completely the opposite. So this digital lock-in amplifier starts by digitizing a large bandwidth of the signal, and then it performs or carries out all the signal processing tasks digitally in real time, which is a lot of flexibility because here you can do different type of processing on the same signal in real time. We exploited this digital lock-in amplifier to do MRI, a parallel MRI acquisition of two different nuclei, namely the proton and the fluorine. The setup looks like that. We have the Bruker system that is used to control the gradients. And we use the digital lock-in amplifier to provide the excitation signal and to do the acquisition, and do that simultaneously for two different nuclei. So inside the lock-in amplifier, this is the block diagram inside the lock-in amplifier. So we have an output channel, a high-speed digital to analog converter that can generate signals with extremely large bandwidth. So you can have one excitation pulse with two frequency profiles that can perform slice selective excitation of two different nuclei. And we have also a high speed, we have an RF input with a high speed analog to digital converter that can digitize large bandwidth up to 600 megahertz. And then we have a real-time FPGA processing, FPGA-based processing unit that can demodulate up to 8 different bands simultaneously. For this, the imaging sequence, we used a gradient echo sequence. where we added also a TTL trigger signal so that we can synchronize the two systems. And for the RF excitation, we used one single excitation pulse that has two RF profiles to do slice selective excitation of both the nuclei. And then when we acquire the signal is a combination of two FIDs of the two nuclei that can be separated digitally in the local amplifier. Of course, we needed to modify the detector, so there are two options. Either we make the coil a double resonant coil, or because the frequencies were quite close to each other, we're talking about 42 and 45 megahertz, we thought it would be easier if we just increase the bandwidth of the coil by artificially adding or lowering the Q factor of the coil. Here's the excitation pulse in time domain. In the frequency domain, it shows that it has two nice sharp excitation profiles at 42 and 45 megahertz with a bandwidth of 3 kilohertz. That's the slice thickness. And the sample we use, the sample holder, is a 3D printed sample holder with two chambers. One is filled with water, and the other one was filled with hexafluorobenzene as sample for fluorine. And these two images are acquired simultaneously with a single RF channel. Okay. The other thing is, if we are talking about whether we are talking about an MRI phased array or we're talking about an NMR spectroscopy array, if all the detectors emit signals at the same frequency, it will not be straightforward to just combine the signals. In this case, we still need kind of a dedicated receiver for every detector. And that will increase the complexity and cost and size of the system. Therefore, we introduced or we used CMOS technology to build this microchip that comprises 8 receive channels. So the CMOS chip, this is the block diagram of the CMOS chip, and this chip, as I mentioned already, it contains 8 receive channels, each of which has a low noise amplifier frequency mixer to demodulate the NMR or MRI signals, and then there is a bandpass filter to suppress the high side bands after the demodulation. And after that, since we can also demodulate every signal to a different frequency band, we can do frequency multiplexing by summing all the signals and carrying them all over a single cable, which can then be acquired with the local amplifier, with a single digital local amplifier receive channel. These are the schematic diagrams of the different components of the CMOS chip, which was fabricated using commercially available 350 nanometer semiconductor technology from X-FAB. And the chip was tested in a high field. The performance of the chip was characterized in a high field system up to 11.7 Tesla to ensure that the performance in terms of linearity and gain of the chip is not affected by the field. And after that, we tested the chip in MRI experiments in high field system, preclinical imaging system of 9.4 Tesla magnets in an imaging experiment, gradient echo imaging experiment. And this is the image from the chip compared to the image from the Bruker. We see that it's capable of acquiring the signals faithfully, even though the noise performance is not as great as the commercial system. We have a drop in the SNR by around 3 dB. In a low field system, we use the chip, but we also use external preamplifiers with the chip, and we could see that with external preamplification, we could enhance the noise performance, and we could get more or less similar quality as commercial spectrometers. And those both experiments show that the chip can work over a broad range of frequencies from few megahertz to around 700 megahertz. And then we can combine effectively 8 channels together and acquire them using one single channel of the local amplifier. So that shows the potential of CMOS electronics in tremendously reducing the complexity of the NMR and MRI hardware. To summarize my talk, as I mentioned, the main motivation was to go to achieve high throughput NMR, and to do that, we needed to tackle several technical and methodological challenges. We talked about parallelization. We talked about miniaturization and optimization as key things to have efficient shims. So shim sets with as little or as few shim coils as possible, but still capable of achieving high spectroscopic resolution. We talked about applying deep learning. This has a large potential in speeding up the shimming process, and so far we have applied it to spectroscopy, but this also can be a good approach for imaging as well. Digital lock and detection also holds great potential because this the ability to digitize large bandwidth and then do all the signal processing tasks in real time afterwards on multiple frequency bands gives a lot of opportunities to decrease largely the number of needed receive channels. Also microcoils, these are important because if we want to pack more detectors in the magnet, we have to make their size smaller, and we need a kind of simple and reliable way of manufacturing those microcoils. And finally, CMOS electronics also show great potential in significantly reducing the complexity and size of the hardware of the NMR and MRI system. At the end, I would like to thank my group for this work, and I would like to thank all the people who contributed and collaborated in the work, and I would like to thank the funding entities, and then I would like to thank you all for your attention. And I will be happy to answer your questions.
Yeah, Mazin, thank you. Thank you for lovely and broad perspectives on what you're doing in your group. I have actually quite a few questions.
Mm-hmm.
So let me start, and then maybe others will join me. I had a question about the deep learning. Actually, I was curious.
Mm-hmm.
Do you need any? So I would think for simple machine learning, you would need assignment or something. And you said that you acquired only some 1,000 spectra.
Mm-hmm.
And then how did you tell the system what spectrum should look like at the end? How does machine know how it should shim it then?
Yeah, that's a very good question. So what we did here is we started from, we use this magnet here to collect the data. We started from a trimmed situation where we have a good spectrum, and then we purposefully distorted that spectrum by applying a distortion along the X and Y and Z.
Mm-hmm.
And then we recorded the corresponding spectrum.
Mm-hmm.
And the idea here is that we then feed this distorted spectrum to the model as an input, and we tell that, okay, whenever you get this distorted spectrum, you should predict those X, Y, Z values as the corresponding values that could correct this spectrum.
Sort of, you use only one spectrum for, like, as a gold standard, and then all the rest were distorted. And machine was trying to predict parameters. to go to
more or less
this.
the same. Exactly. So the model takes the spectrum as an input, and it should be able and takes the correct shim currents XYZ as the label that corresponds to the spectrum. So whenever he gets a distorted spectrum, it will kind of predict, okay, if I get this spectrum, then I know that, okay, I have to predict X, Y, Z like that. So then when you apply those values that are predicted by the model to the machine, you get to the right shimming condition.
And sort of you need only one reference spectrum at the end. It was sufficient.
No, once the model is trained, I don't know what you mean by reference spectrum.
Like the good shimmed.
No, we don't feed that to the model. No, we of course we need to start from a good shim so that whenever we give the model the labels XYZ, those are the labels that brings any unshimmed spectrum to the correct shimming condition.
Okay.
I don't know if that was clear, but yeah, basically we start from a good shim and then we start distorting the shim with kind of a uniform distortions as you see in this grid here.
Yeah.
And then for every case, we give the distorted spectrum as an input to the model and tell him that the correct XYZ for that spectrum is this number or this configuration.
My only question was here it was like about 9,000 spectra, and all 9,000s were distorted, and only one was well shimmed.
Yeah.
Yeah.
Exactly.
And maybe hello, and thank you so much.
Go on.
Hi. Maybe one connecting question. And was it all on the same sample or different samples?
Here it was all water. It was the same sample.
So you kept the sample inside and you varied the shims. You didn't move the sample in and out or so.
Yes, yeah, that's a very, very good question. Of course, I mean, we noticed that if you change the sample, then the shimming also is disturbed. So here we ensure that we didn't move the sample, we didn't rotate it even when it's inside the magnet. And then we collected the whole data set over, it takes a week to collect that data set. And and then we collect it once and then we train the model.
Thank you.
You're welcome.
I have few more questions, but one is rather educational. Because on slide 5, you showed how an NMR signal can be sort of calculated from B1 and M0. And I was curious about notations. Like here. If I would think that so B1 and M0, is it the scalar product or it's a vector product? Because so it should be scalar because at the left side I see a scalar product. My problem simply is that B1 is changing along transversal plane, right? And M0 is here should be orthogonal.
It's a vector product. It's like
it's the vector, yes. But the product of orthogonal vectors is 0. I was just curious how to understand this notation.
Mistake. No, it's a vector product. It's basically the.
No, but vector product cannot be correct because then it should be a scalar. So the vector product is a vector.
Yeah, I mean, here's it's a bit confusing here because we are only concerned about the amplitude, not the directions. But effectively, if you want to trace the evolution of the magnetization, it's basically the vector product of the B1. It's like the Bloch equations, right? It's the vector product of the field and the magnetization. And then you have all the components of the magnetization. But here we are only concerned about the strength of the signal. So it's effectively we're just the magnitude of the B1 and the magnitude of the magnetization.
Yeah, but so here it's assumed M0 is along Z-axis or along, for example, X-axis. So it's
No,
already
that's when it's excited, right?
so it's after excitation then. Ah, but if it's after excitation, then it's all correct.
Yeah.
Yeah. More question from
Yes,
our audience?
yes, I have a small question. Did you, maybe I missed it out, I was leaving in between. Um, did you compare the SNR of the sort of broadband lock-in amplifier and the standard NMR amplifier?
That's a very good question. Yeah, actually, we did that. Unfortunately, I don't have the slide. Yeah, the results are very comparable because it might sound a bit counterintuitive because, of course, if you demodulate the signal in the analog domain first, go to that. If you demodulate the signal in the analog domain and and then you can use faster ADCs there when the signal is in the baseband. But because of the oversampling, the insanely high oversampling ratio offered by the lock-in amplifier, you can still do decimation or moving average of the oversampled data to further improve the SNR. And at the end, you will see that it's very comparable.
So you did an experimental comparison and the SNR turned out to be similar.
Yeah.
Okay. And yeah, okay. Thank you.
You're welcome.
Yeah. May you comment? So I think Bruker from Neo Console, so at least what I understood from the advertisement, they say that they measure it now directly without demodulation.
Okay, that's
At least maybe I'm wrong, but it's what allowed them to. So they say they increase the SNR this way because then they have less electronics involved.
okay.
But I thought maybe you know better because you're in the topic.
Yeah, to be honest, I don't know about the new Neo spectrometer. I know, for example, for the magnet we use, the preclinical one, that's 45 megahertz. That yeah, they're doing analog demodulation first, and then they digitize, I think, at 20 megasamples per second when the signal is down converted first. But it could be, yeah, for that. It makes sense actually because now the advances of semiconductors and you can have ADCs with high bit resolution at very high sampling rates, and then everything becomes easy once you digitize the signals, right? And then, yeah, you can also do multiple parallel processing on the same data set in real time. So
What I have in mind is that, or what I remember is that maybe before they downsample to a lower frequency and maybe they downsample now uniformly to a higher frequency, or they had before two analog stages and now one, but I'm not sure if they do really megahertz sampling directly. I'm not so sure if megahertz or even gigahertz sampling can be done directly for Bruker in the Neo console. But I remember as well that they have now an improved digitalization step there.
Okay. Yeah, you're right. Might be not just to skip one off because typically they also do some kind of super heterodyne demodulation. So by having multiple frequency conversion stages to also suppress the images of the frequency band, but it could be I have no. I'm not really sure about the new architecture of the new systems, but could be that yeah they're skipping one frequency conversion step. Yeah, gigahertz sampling might be a bit tricky. Yeah, because yeah, as far as I know, there are already you can get like ADCs up to 50 gigasamples per second, but then the vertical resolution is not great. Of course, yeah, you need a very complicated processing afterwards, which might not be worthy.
Yeah.
Maybe then I'll just ask two last questions
Mhm.
about the coils design. You said that you need to sinter your silver coils
Yes.
after printing them. I was curious, maybe you just explain how sintering works, because the silver is not doesn't melt at 200 degrees, but then it still spikes, or how is it melt? Why is
Um,
it enough?
that's a good question.
The melting point is around 1000 Celsius.
Um, yeah, I just need to go to, yeah. Um, that's a good question. I think it would still be particles because normally we used to do electroplating after the sintering, so it will be kind of a rough surface of silver particles, and then we electroplate it with copper. Um, yeah, but um,
On some images, some other slide, maybe 15 or so,
yeah.
somewhere you showed here this microscope, this the images of the, I think the C is the
Mm,
coil. It looks very smooth to me, but I wonder maybe the particles even smaller.
mm. Yeah, actually, that's a good point. I'm not sure if it's 220. I have to check again the numbers. It could be that it's not this. Yeah,
Mm.
yeah, that seems too low for the sintering process, actually.
Yeah, I don't know. Maybe just you remove the glue this way. I just was wondering.
Yeah, that's probably just to dry it and cure it, and probably the sintering is at high temperature. I have to check the number again.
Yeah, thank you. And the last question, maybe a few slides after it, was these butterfly coils.
Yeah.
I'm not, so I know how the simple RF coils are working, but maybe you can explain how current flows in these flat coils. Maybe,
You mean the conventional
yeah.
supply?
For example,
Yeah, it's really simple. You can think of it as a single loop coil. So you have two planes. For example, you feed the current. Do you see the mouse?
yeah.
Maybe you can just use a laser pointer. So you feed the current to the, for example, to the upper layer, and then of course this is a wide copper layer. So the current density here is low, and as the current goes through this narrow part of the copper plane, the current density becomes higher, so you have a stronger field. And then the current density becomes again lower, so you
Mhm.
have lower field under this plane. And of course, the here typically there is a kind of a via connecting this upper plane to the lower ground plane. And the
Mhm.
current comes back from the bottom ground
Mhm.
plane. And then you can place just simple tuning and matching capacitors on this side, for example. So it's like a piece of wire. You
Mhm.
can think of it as a single loop coil where one part of the wire is made narrow so that you have a larger current density around that wire where you can put your sample, and then your coil is more sensitive to the sample at that region.
Mhm. Okay. Thank you.
And when we replace that, because now you can't, for example, you can't increase the sensitivity of the coil unless you make this part narrower. But of course, then your sample volume becomes smaller.
Mm.
Or you have to make this part wider, but then you have space constraints.
Mm.
The solution for that is to replace this part with a butterfly coil where you can add, if you have thin wires, then you can, of course, add more loops, and
Mm.
then you will be able to increase the magnetic field.
Mhm.
I have one question to this image that you show. So in the butterfly coil, in the zoomed area, I would assume that those are inductively coupled coils, so they're closed loops. Is that correct? But it looks like there's an open end in the center. If you go, yes, there.
Here, no,
Yes.
yeah, actually, the current, they are connected to the copper, to this plate, to this copper part, as you see here.
Okay.
And then the current splits here and then flows in opposite direction.
Gotcha. Okay.
And then both are connected
So
back to
the light,
wrist.
yes, the light yellow is the conductor. Yes, of course, as you showed it everywhere else. Yes, I understand. Thank you. So it's not inductively coupled, it's just really the same conductor.
No, it's electrically coupled.
Yeah. Yeah. Thank you.
You're welcome.
And in this, I'm sure in this graph, you also discussed that the butterfly coil is more sensitive. But to me, it looks like there is some trade-off because it's also more inhomogeneous, right?
Yes, yeah, that's one of the drawbacks, as you can see here, also from the notation spectrum. It's not as uniform as the conventional strip line. That's one drawback. So if you care more about sensitivity and shielding, like here, for example, you can completely suppress the sample outside the sensitive region. So if you care more about RF coupling and unwanted sample suppression, then it will be superior. If you care more about the uniformity, and that's typically what spectroscopists prefer more is the uniformity of the B1. Then of course the conventional strip line wins.
And your samples, are they longer than sensitive area usually, or they can be smaller?
Yeah, it's typically you have to make it longer, because otherwise, if you just make your sample only to the sensitive area, then you have the air sample interface very close to the sensitive region, and then you have the susceptibility jumps, and then you have problems in the line width, the shimming. So typically, you just use a long capillary, so that you try to make the sample air interfaces as far as possible from the sensitive region. And so if your coil is more blind to the region outside the sensitive, then it's better, because it simply doesn't care about how the sample looks like here.
Yeah.
And maybe a not so important question, but how did you quantify the SNR in B? Did you use the integral, or did some fitting there?
Here it's integral.
Yeah. Okay. Thank you.
Okay.
I guess one can do something like Shigemi tubes design with the glass, Shigemi tube. It's the tubes with the thick bottom, so that the glass has a susceptibility match for water. And then you won't have the B1 problems anymore.
Yeah, that's a good point. That can be also an option.
I guess that's again. Thank you, Mazin, for live presentation. I have no more questions, and I don't see questions from the audience. Then, with this, I will close the meeting and wish everyone a very pleasant evening.
Thank you very much.
Good.
Thank you.
Take care. Bye-bye.